The overall goal of this procedure is to provide a detailed methodology to study the horizontal transmission of DNA in Staphylococcus aureus by addressing the three main transfer pathways: conjugation, transduction, and natural transformation.
This method is about horizontal transfer of antibiotic resistance genes in human pathogen Staph aureus. The main advantage of the techniques introduced here is that we can test three major modes of transfer, including recently discovered natural genetic transformation. Demonstrating the procedure will be Thuy, a PhD student from our lab.
To begin the experiment, prepare 5 milliliters of overnight culture of the donor and the recipient bacteria in tryptic soy broth, or TSB medium with and without 32 milligrams per liter chloramphenicol, respectively, with shaking at 37 degrees Celsius. The next day, adjust the optical density of the overnight cultures to 1 with fresh TSB medium. Mix 0.5 milliliters of the donor culture with 0.5 milliliters of the recipient culture, and add 1 milliliter of phosphate buffered saline, or PBS, to the mixture. Then, using a vacuum pump system, transfer the mixture of bacteria onto a 0.45 micrometer filter membrane. Place the filter membranes on a sheep blood agar plate. Incubate the plates at 37 degrees Celsius for 1 day. Take out the filter membrane from the plate and suspend it in 10 milliliters of PBS. Vortex the mixture for about 1 minute to collect all the bacteria attached on the membrane.